When we think of genetics, a lot of the time we think about how there are differences between different people, different organisms, even different branches of living species. So, if you recall all the way from evolution, that mutations are what helps organisms have greater fitness or less fitness out in survival and reproduction. So let's take a look at what kinds of mutations or variations in genetics can occur and how they occur. So there are two kinds. There's chromosomal mutations and gene mutations. The first one is chromosomal mutations. That's when some part of the structure of the chromosome or the number of chromosomes change. So for instance, person with Down syndrome, instead of having 46 chromosomes, which is 23 pairs, instead will have 47. And so what that looks like is that on chromosome 21, instead of having a pair, notice everyone else here had a pair. Chromosome 21 has 3 copies of that chromosome. And so that is what leads to the disorder we know as Down syndrome. So, a little extra information, it generally affects 1 in 700 people. It can change a person's phenotypes, that means what physical characteristics they have, and it can also affect other organisms other than humans. So, the other type of mutation is called a gene mutation. And so, that's when we have a change to the actual bases on our DNA. So that's our nucleic acids, and something changes there. And when that happens, it can cause abnormal proteins to be created. And basically, if we create abnormal proteins, we are not able to use those because they're not the ones we need. And either they just won't be absorbed or there can be other problems occurring from it. And so the two types of gene mutations are point frame mutation and frame shift mutation. So let's take a look. Point mutations is when a single base or a single nucleotide, you can use either word, is changed. And that can happen at any point because our DNA is replicating. Think about your skin cells, right? There's always new ones coming up. And so anytime that there is a replication, there is an option for a potential mutation, like an accident. And so if we look at this first example, ACA becomes ACT, so that last A was accidentally replicated as a T instead. And so what that means in this specific case is that actually ACT will get transcribed into RNA as UGA, and then that's a stop codon. So anytime you see this word stop, what that tells us is that it is going to stop the making of any amino acids and therefore the stopping of any proteins. So instead of this just being one little spot on our amino acid chain and creating a lovely protein, suddenly we're being told, nope, you're done, you're done, you're done. And so the actual protein never gets made. Okay. Now, the other type of gene mutation is called a frame shift mutation. And so if we take a look at it, frame shift, think about that shift word. That means we are shifting or removing or adding some kind of nucleotide into what's already there. So here's our example. I used words in this case to make it a little clearer. So let's say we had this phrase, the cat ate cake. And in a frame shift mutation, something would change in here that would kind of skew everything. So let's take a look at this new version. The, and then ATA, and then TEC, and then AKE. So something got set off and everything got moved over. And so the thing that happened is that this first C got unfortunately deleted. And so now everything shifted over by one. And so now instead of saying the cat ate cake, we have this nonsense phrase. And so the same thing happens in DNA and then in RNA to create proteins. Okay. So now when we're looking at what these mutations can do, remember that there's lots of mutations that provide increased fitness to the organism, right? That's how natural selection works. If that mutation provides an increased advantage to either survival or reproduction, then that individual has a higher chance of reproducing and then that specific mutation getting passed along. Now, on the other side, though, when that mutation is not advantageous, we can have something called genetic disorders. So there are 3 different ones. Let's take a look at them. So the first one is called a sex-linked trait, and that means that where that mutation occurred is on the sex chromosomes, which are X and Y. Okay. And the reason it's important to know if it's a sex chromosome or a body chromosome, we're going to talk about in just a second. But because we get different amounts depending on the sex of the individual, will depend on whether that trait is going to be a carrier, whether it is going to be actually present, and that individual will notice it in terms of some kind of change in their body, or if not. So those are things to kind of keep in mind. So let's take a look at an example. So reminder that sex is a biological term that is specifically referring to the DNA sequences of um, for cisgender males as XY and for cisgender females as XX. That does not mean those are the only options. There are many more complications to sex and also gender. But in this case, in terms of how we're thinking about it for a living environment, cisgender males going to be considered as XY, and cisgender females will be considered as XX. Okay, so if we take a look here, because cisgender males have one X, if there was some kind of mutation on the X chromosome, because they only have one instead of two, like the cisgender females, there is an option that whatever change happened on that X chromosome is going to affect them really hard because that's the only one they got. So there are a few sex-linked genetic disorders that get passed along that way, and they can make it a little bit. We're going to take a look right here. So easiest way to think about it is because of the XY, if the cisgender male child of a cisgender female has that genetic trait, then that cisgender male child has a 50% chance of inheriting that gene. So it's pretty high. Usually we think about inheritance of disorders as being really small percentages, but because they only have one X, that percentage goes up really, really high. On the other hand, if we look at a cisgender female child, it's going to have a 50%, which is also a pretty high number, right, of just being a carrier. And so a carrier means is that they may have that change on their gene, but because there's 2 of them, let's say this one has that change, there's a second X chromosome right here that allows the option that they may not even experience any changes from that gene. Okay. So we talked about sex-linked, so now we're going to talk about autosomal. So autosomal, soma means body, auto means yourself, and so every human has 23, generally, pairs of chromosomes, and one of those pairs are the sex chromosomes that we just talked about, right? There are lots of variations. These are just the most common. This does not mean they're more normal or less. They are just the most common for the sex chromosomes. The other 22, right, because there's 23 pairs, the other 22 are called autosomes because they are on the body. They're body chromosomes. They are not sex linked. Okay, so these are called body. There we go. And so anything that says autosomal are going to be on the non-sex linked chromosomes. Okay, so for example, sickle cell anemia is an autosomal genetic disorder, and it impacts hemoglobin, which is the cell that is the chemical in your blood that carries oxygen and makes it turn red or that purpley color. And when somebody has the genetic marker for this, it impacts how a certain amino acid and therefore a certain protein is made. So instead of having the red blood cell look like a disc shape, like a frisbee, it kind of looks like this shape, which is also called like a sickle shape. And so that's where this name comes from. Sickle cell anemia is from that shape. All right, so as we are thinking about this, you're like, how do we know what this is and how do we test for it? So there are two ways to test for it when in utero. And so this is not for the parents, this is more for the offspring. So one option is to use amniocentesis, and so that is to get a sample from the amniotic sac that surrounds the fetus, and there are cells in that amniotic sac that are from the fetus, and so they can take a sample and then take a look to see if there are any very obvious genetic markers that would help them maybe diagnose Down syndrome because of that trisomy, or a few others that are a little easier to spot right there. And so the other one is called karyotyping, and that's when it's a blood sample, and then they actually get all of the chromosomes lined up and can take a look to see if there are any obvious abnormalities, if there's any changes in shape or length or size that would help them easily distinguish genetic disorders. There are ones that are much more complicated than just looking at chromosomes under a microscope, but they tend to look for the more obvious ones this way. And so here's another image real quick. When chromosomes are together instead of as a pair right next to each other, they get attached by this thing called a centromere, which is right here. And that can also tell us some information about the chromosome itself. Is it really high up? Is it really low? And so we're going to take a look at the next slide real quick. And if you notice, here's 1 through 12, and then 13 through 22, and then the X and Y chromosomes. And so this is part of karyotyping where they can kind of band it out and take a look at where that centromere is. It's right in the middle right here. It's a little higher here. And then take a look at these banding to help them figure out if there are any obvious genetic changes that they want to be aware of in the offspring. So that is where we're going to pause. If you have additional questions, feel free to reach out.